Okay, good afternoon, everyone. Let's start. I hope you have already, everyone has already seen the video that I have already posted 2 days ago. It's about, it's important for rDNA in medicine, primarily because in several ways. Its first thing is about as an application of PCR, you should see this is that is one way of seeing, and if also as a molecular diagnostic tool. These are all things that you will come under molecular diagnosis, molecular genetic diagnosis. Say if we want to know if a person has a kind of genetic condition, it is also involved in cancer research and many other things. It is about trying to find out if an individual has any kind of mutation, and mutation is any change in the sequence, right? And the mutations could be insertion, deletion, and all those many others, insertion, deletion, substitutions, and so on. If there are large scale insertions or deletions, we can figure it out using techniques like Southern blotting and other things. Even karyotyping can also be used. It should be larger fragments of deletion or insertion. But if it is very tiny deletions or changes, something like point mutations are um, that is one or a few bases. The change is in one or few bases. Then how to deal with it, how to diagnose it. Obviously, we have to go for sequencing. Sequencing is the one that it is the ultimate diagnostic tool. It's a kind of, for every discovery of a point mutation, it happens through sequencing only. There are other tools or follow up of sequencing. So once we have had the information about a point mutation, and now we have 1,000 people and we want to see who all have it or not. Sequencing all these, the region of all these people is actually it's not cost effective. Sequencing is expensive. Okay. Um, it is easier and faster to do to further ascertain a particular point mutation if it is there or not in a large population. PCR based techniques are very well used. You can do the multiple samples at a time, and it's easier and it's faster too. Okay, that's why. So once a point mutation is characterized using sequencing, people then design experiments based on the mutation, and then they will try to do experiments. So these are some of the techniques that we are talking about. There are three things that I've discussed. You can have a large number of variations from what is already there. First thing before I go, we need to get the terminology right. Alleles are alternate forms of a particular gene. Okay, gene is there, and say for example here A1 and A2 I'm saying as two different alleles. This is the gene. The majority of the sequence is same except an insertion of this part, about 100 base pairs. Okay, here one line indicates a double-stranded DNA. Okay, um, so here what I'm trying to do now is I will try to describe again what we have shown in the video, and then we will follow up with a problem based on this type of question. So there is a DNA which has A1 and A2 alleles. A1 is about 300 base pairs. If I used the primers F or F and R, that is forward and reverse, it will give a size of about 300 base pairs. If it were A2 allele, because it has a 100 base pair insertion, the length of the band that amplified DNA would have been the 400 base pairs. So we have isolated DNA from different samples, different people. Okay, and then we have isolated, and then what you see here, based on the banding pattern, you can figure it out. If it is giving only 300 base pair fragment and no 400 base pair, because a majority, all mammals are diploid. Diploid means they should have two sets, right? So if there is only 300 base pair fragment and no 400, it must be A1 and A1, right? If it were so this is A1. If it is only 300 base pair fragment, with the same logic, if it is A 400, it must be A2, and the second one there is no 300, so it must be A2 as well. So basically we are deducing the answer from it. Okay, and here is there is a 300 for the person third individual. There is a 300 base pair fragment and a 400 base pair fragment. From 300 base pair fragment, you can say that it is A1 is present, and from the 400 base pair band that you see, you can say that A2 is also present. Therefore, say if I call these as X, Y, and Z individual names, X is homozygous for A1 allele, Y is homozygous for A2 allele, and Z is heterozygous. It has one A1 and the other one is A2, right? Typically, what would happen? You would do all those things. You will see the band pattern, and then you will be able to describe these things. Now, I hope you all understood. While I'm discussing, if you have any questions, I would like that you question me in between when the explanation is happening, or else it will be nicer that way. This is supposed to be an interaction, and it's a kind of tutorial for the pre-recorded video. There has to be an explanation to things. If you are lost somewhere, please do ask so that your reasoning is good. My goal is that to improve the reasoning for this kind of purposes here. So here is a question based on what is done. Everything is same except no, almost everything is same. Only thing you need to check is what is the genotype of A1 and A2. A1 is about 400 base pairs, and A2 has a deletion of 100 base pairs, so it would have been 300 base pairs. If it is when you use two primers and amplify, a fragment forms that is called as amplicon, like replicon, like that. It is called an amplicon when you use two primers and get a fragment. So the size of the amplicon is about 300 base pairs for A2, and A1 it is like this. Okay, that is the setup that we know of information. We are using primer P1 and P2 just for these. Okay, I will call this as L2. So we have three individuals. They are L, Q, and R. And the DNA, we have isolated their DNA, and we performed PCR with P1 and P2. This is the band pattern that we got. So now you have to determine what is the genotype of LQR. On the paper, draw this diagram and try to determine what it is. And those who are finished, you can answer. I'll give you 2 minutes before you people, anybody answers. It is quite simple, I know that, but we will take anybody who may have difficulty. We'll carry on with all. So please quickly solve this, and always use the rationales of, reasoning properly, so that it will be nicer. And I always want you to remember the fundamentals of electrophoresis. The smaller ones will travel faster, right? And the larger ones will travel slower. I would also, I would also like, sometimes I might give in the question paper, if there is anywhere we might have a question paper. we may have. If it is there, I may give a gel diagram and tell you to pick all the mistakes in it. Okay, and then you're supposed to do those things, and that might get that means you have to be very critical about every aspect that is given here. If there is any incomplete information or if there is misinformation, you have to train yourselves so that you will not have you will not get any faulty information into your head ever, not just this, in any file. So some people say this, right? Science, what is science? Is celebration of doubt. That means you're supposed to doubt it and try to find out what is find out if there are any questions, any mistakes in it, and some critical aspects. That's what one is supposed to exercise. So I hope you'll remember this and try to inculcate that kind of attitude, so that we question things and we see if today I'm saying something, and nobody should blindly believe that. You should be able to question the information that is coming out of it and try to use your reasoning skills or explore more and then find out. So what is, I think I'm getting some answers already. So I want to know what is L? What is the genotype of L? That is how we should say. L is heterozygous. 300 base pair says it has A2, right? And 400 base pair fragment band here shows it has A1. So it is heterozygous. And Q is 300 base pair. It shows a 300 base pair fragment, but there is none here. So it must be A2 and A2. And in cases like R, let's see where we see only 400 base pair, therefore it must be only A1, and then finally we will all read it as individual L is heterozygous for A1, A for A1. So I hope you understood this, and this is pretty simple. I would want you to appreciate that and that way. So the next one is we discussed about this one. So in this we restriction your digestion EcoR1 site GAA-TTC. It is a substitution, means there is one change. You should realize that in the first assignments you to try to describe the mutation in all possible ways: transition, transversion, point mutation, substitution, all those things. And that is why first assignment is the key to your success in the rest of this one, because you learned molecular biology a while ago. Anyway, so here we have two different alleles, A1 and A3. A1 has a restriction in site GAA-TTC in this location. That's EcoR1 site. I want to mention this before I forget. In majority of the slides for the assignment, second graded assignment, I gave the rubrics. In one mark is for how you are writing the organism names or restriction, the conventions, are you following or not? So many people, I told EcoR1, if you are typing it, you should have used these three alphabets should have been in italics. I have found so many people that haven't done that, so there will be a problem with that. And in some cases, say for example hint 3, somebody wrote like this. This is wrong. It should have been hint 3. These three should be italics. This is the strain name and Roman number. It is just to recall, clear, make you aware of it so that you will be able to focus on that. Okay, so here is eco R1 site. Shweta, you have a question?
Yes, sir. As you said, for
Okay.
the italicizing the first three letters, I just googled up because
Yes.
I went through few websites. In those particular website, they did not italicize. So when I googled it, it said according to 2003 convention, the first three letters should be normal and it shouldn't be italicized, sir. So that's the reason why, yes,
Oh,
sir, it has been, it just said that. So that's the reason why I did not italicize
uh,
the first three letters, sir.
where did you get this information?
I can send you the link, sir.
Yeah, please post it here so that everybody will be able to see
Okay, sir,
that.
sure, sir.
Typically, E. coli, if you're writing, organism should always be utilized. Okay, so this E. coli is taken from eco is taken from there, so they are saying there it was italics, but that's all right. Then if there is, you'll get the benefit of doubt. Okay, but I hope it's a standard one. The organization that says that. I wasn't aware of it. Thanks. Let me find out first. Okay, so if you are having an eco R1 here, and because of a substitution, A is now replaced with T, and therefore you have a transversion. This is a purine replaced with pyrimidine, right? And now, if A1 can be cut into 100 base pairs and 200 base pairs, but if you eco R1, if it fails to act on this, so it cannot be cut and it will remain as 300 base pair fragment. Now, if I got two bands like this and we did not get, so there are three possibilities here. Yeah, there are two possibilities. Either you will get 300 base pair fragment or 100 and 300 base pair fragment, like this. So you have in the case of X, Y, Z, we have isolated DNA from them, and then we did PCR followed by restriction digestion. We get 100 and 200 and no 300. Therefore, it must be A1 and A1 because there is no 300 band here. In case of Y, you have only 300 base pair band. That means 100 and 200 are not there, though, so it must be homozygous for A3, A3. And in case of Z, you have all the three bands. So these two must mean it is A1, and this one must mean A3. So Z or Z is heterozygous for A1 and A3. Okay. Yes. So we will try to solve a question, and after that, I will take up this link and I will check meanwhile. Sorry, not in this part. So quickly, I would like you to solve this. I have made a question here. Look at the question carefully and critically so that you can see the information, because if I describe everything again like the previous one, it'll be difficult. So A1 and A3 here, the scenario is a little bit different. Different in the sense you have GGA TTC, and then there is a mutation in A3 substitution, because of which something has changed. So you decide what to do with it. You would have taken the DNA from people X, Y, Z, and then you did a PCR, and then restriction digestion using eco R1. And then we got banding patterns like this. So this should be 200, this should be 300, and this one is 500. Okay, so you please try to. I think this is pretty straightforward too. As soon as you're done, please post it in the comment. Yes, uh, I think it is done, right? So quickly, with this, X has given 200 and 300. Okay, first thing is echo R1 will be able to cleave here, but not here. So this in because of a mutation, a restriction site has formed. If you look at the problem, that actual one we are talking about, because of a point mutation, there is a loss of restriction site in this case. For the next problem, I just changed it a little bit, where because there is a mutation in this for G here, we have a we have now a restriction site has formed, echo R1, which was otherwise not. So A1 would give 500 kb, 500 base pair fragment, whereas A3 would give 200 and 300 if treated with echo R1. So X has given for 200 and 300. So it must be A3 and A3. Y has given for A3 as well as A1, this one. And for Z, it's only 500. Therefore, it must be A1, A1, right? That is how we can identify the alleles. So let's check out the last one. In this one, you have to do a lot of work. Um, we will see. It is called as amplification refractory mutation system. I like to just call it as allele specific PCR, but this has something to do, something interesting about it, right? It's about everything is in the primers and your ability to design primers. So here, the difference between A1 and A4 is this one base, right? Here it is GACTA, and here it is GACTG. Okay. So in that sense, you can, the primer that you are using here, primer 1, cannot be used here because the DNA polymerase will not be able to recognize this if the base pairing is not there between the ultimate base of the primer with that of template, then DNA polymerase will fail to recognize it. Please also remember that when anytime we are doing a PCR for this kind of things, if we had a polymerase which has 3' to 5' exonuclease activity, what would it do? It will think that this one is wrong base. I mean, not that it will really think, but just for sake. It will identify that this is a mispair, and it will use 3' to 5' exonuclease activity and remove off the base that is not properly base pairing, and then it will start continuing the polymerization. We don't want that. Okay, so we should have chosen a DNA polymerase such as Taq polymerase, for example, which is normally, of course, used, that does not have 3' to 5' exonuclease activity. Okay, keep that in mind. For this kind of experiment, you need that. So if I use primer 1 and primer 2, if A1 is present, it will give a band at 300 base pairs, such as this one. If I use primer 1 and primer 2 on A4, it does not give a band, right? Because of this primer is not matching with that. Okay, so you should have used A3. If we use A3 and uh, primer 3 and primer 2 on an A1 as template, it does not form a product either. So this system can be used to identify if there are if they are homozygous for A1 or homozygous for A4 or heterozygous and so on. So in this case, we use two different primer sets, like 1 and 2. What will it give? If it has given rise to, if a 300 base pair fragment is found, that confirms that it is A1. If there is no band here, then it means that it is not A4. Therefore, it is likely that A1. Okay, with 2 and 3, if it is not giving, then because this is there, it must be A1 as well. In A4, in the other case here, we did not find a band with 1 and 2, primers 1 and 2. So it is not A4. Sorry, it is not A1. And there is a band here. So it must be A4. Therefore, the other one is also A4. So the person, second one, is homozygous for A4. And A1 and A4 are they have a band for in both the case with primer 1 and 2 and primer 3 and 2. So it is A1, A4. Any questions here? If there are no questions, we will go ahead. I hope you understood this. Okay. Yes. Now you will have a lot of work to do. Please, you are supposed to design the primers first thing, and then I want that A is, I'll put as A is X2, X2, and B, person B, individual B is X1, X2, and C is X1, X1. So first thing you need to do is design. So every experimental setup that we have done so far is same here also. We will isolate DNA from people, and you have designed the primers. You have to design primers that are specific for X1 allele. You also have to design primers that are specific for X2 allele. Then that is design primers and perform PCR. Followed by you will do a PCR primers. We'll call them as primer 1, primer 2, like that. We'll call label your name primers as primer 1, primer 2, and primer 3. Okay. So based on that, you will have two different sets of PCR reactions, and after that, you have done electrophoresis. So when you have done that, I have given the what is the genotype. I want you to draw the gel banding pattern. Okay, please quickly do that. And Once you're done, once everybody is done, we will have a quick discussion and then we will move on. When you're done, please design the primers. You have to do it on paper, take a picture of it, and you should be able to upload it soon. Does anybody have any questions on these? And Shweta, you are right um, about the denomination. I mean, how the conventions of writing restriction endonucleases. Thanks for the information. So I
Okay.
will post the link to everyone. Maybe they can also check it out.
Okay, sir. Thank you, sir.
Yes, thank you. Also on Wikipedia, I'm posting the Wikipedia link. Just go there and you can also have a look at it. The reference is also given, a link to that uh, in the on the Wikipedia page. Okay. Is everybody done? So, well, yes, Ketir Balan, you have something to say?
No, sir.
Okay. I also want you to have a look at something else. If we had the typical, if we had the typical exam paper for, sorry, this was last year's first CAA. It was an easy question paper. I think I would also actually wanted that you people do this, but we will postpone it to some other time. CAA 1. Any questions with that? By the way, if you tell me, I will be able to think of those. Okay. So we have 20 minutes. I would quickly go through that. and then see. It is isochordamers, right? First CAA. So far, we have finished a few certain things, and we would want to. I want you to check this. We will want to review this so that we will be able to discuss. Do you have any questions, or do you want anything to be discussed from vectors? I would want that you should have checked it once again, and then we will discuss that. Okay, right now I want you to tell me quickly. We'll go through some of the questions so that it will be a good review. What are isochordamers? Isochordamers are those restriction site enzymes that give rise to um, identical overhangs, right? Then explain the process of homopolymer tailing. That's too easy and straightforward. Terminal transferase, it's DNA independent DNA polymerase, and then it will just add 2-3' overhangs. Explain the rationale for choice of clinofragment and not DNA polymerase 2 in to polish the ends of DNA sequences using sequences used in cloning. Sorry, DNA polymerase 1. This one is DNA polymerase 1. DNA polymerase 1 is special enzyme. What is the difference between clinofragment and DNA polymerase 1? DNA polymerase 1 it is a polymerase. It has 3' to 5' exonuclease activity. It also has 5' to 3' exonuclease activity. So if you want to fill the gaps in a DNA in a overhang, we should use clinofragment so that it will only fill and not eat away any other extensions that are present on any of the ends. That's why DNA clinofragment is better for filling up. Describe the application of alpha complementation in selection of recombinant DNA clones. Alpha complementation is with blue-white screening. In blue-white screening, there is on LexZ PUC18 like plasmids, we have LexZ prime, which is a part from say until 51 amino acids in the top, which produces a um, region, no, which produces a domain that is alpha domain, and the host strain would have produces the omega domain. These two, when present together, they can function as beta galactosidase enzyme. That is, alpha component is complementing the omega component to produce a functional a functional enzyme. So that is alpha complementation used in blue-white screening. Can you design cloning primers for this? Oh, what I will do is I will stop here, except I want you to, on the same paper that you're doing, I want you to write answers for question number 5 and question number 6. Please do that, and that's the end of the class. Meanwhile, if anybody has any questions, I'll be happy to answer. Yes, please. Anybody?